Hello and welcome to the Neuron Regeneration Podcast and show. Thank you so much for joining me. My name is Jared Carter. I deeply appreciate it. And today is an abstract reading episode. These are designed to allow anyone interested in neuron regeneration research to stay up on the most recently published papers for neuron regeneration, neural regeneration, or nerve regeneration. I'm going to cover 5 different papers today, just looking at the abstracts. We have 2 looking at the effects of exercise or movement training on spinal cord injury, along with other interventions. We're going to be looking at exosomes and their effect on the guidance of axon regeneration, metabolic reprogramming using nanofibers. I believe that's also in SCI models, and then a brain glue that's being used and having really significant results in rat models, or I think mouse models, of post-ischemic stroke. So some interesting stuff. Let's dive right in. And if you want to access any of these papers, you can go to neuronregeneration.com/8, the number 8. Okay, first paper from Burns and Trauma by Ying et al. Title is Exercise Therapy Facilitates Neural Remodeling and Functional Recovery Post-Spinal Cord Injury via the PKA CREB signaling pathway modulation in rats. Abstract. Background. Neuronal structure is disrupted after spinal cord injury, causing functional impairment. The effectiveness of exercise therapy, hereafter ET, in clinical settings for nerve remodeling post-SCI and its underlying mechanisms remain unclear. This study aims to explore the effects and related mechanisms of ET on nerve remodeling in SCI rats. Methods. We randomly assigned rats to various groups: sham-operated group, sham-operated plus ET, SCI alone, SCI plus H89, which we'll discuss in a moment, SCI plus ET, and SCI plus ET and H89. So lots of different groups here, which I love. Techniques including motor evoked potential, video capture and analysis, the Bezo-Beatty-Brescian scale, Western blotting, transmission electron microscopy, hematotoxylin and eosin staining, Nissl staining, glycine silver staining, immunofluorescence, and Golgi staining were utilized to assess signal conduction capabilities, neurological deficits, hind limb performance, protein expression levels, neuron ultrastructure, and tissue morphology. Wow, lots of groups and lots of things that they looked at. H89, an inhibitor that targets the protein kinase A, or PKA, CAMP, response element binding, or CREB, signaling pathway, was employed to investigate molecular mechanisms. So in short, that's going to be the PKA CREB signaling pathway. Okay, results. This study found that exercise therapy can reduce neuronal damage in rats with SCI, protect residual tissue, promote the remodeling of motor neurons, neurofilaments, dendrites or axons, synapses, and myelin sheaths, reorganize neural circuits, and promote motor function recovery. In terms of mechanism, exercise therapy mainly works by mediating the PKA CREB signaling pathway in neurons. Conclusions. Our findings indicate that 1. ET counteracted the H89-induced suppression of the PKA CREB signaling pathway following SCI. 2. ET significantly alleviated neuronal injury and improved motor dysfunction. 3. ET facilitated neuronal regeneration by mediating the PKA CREB signaling pathway. And 4. ET enhanced synaptic and dendritic spine plasticity, as well as myelin sheath remodeling post-SCI through the PKA CREB signaling pathway. So, wow, I mean, exercise therapy and movement therapy for decades now, we've seen that adding movement training or exercise, and in many cases, these are scenarios where the subject or animal cannot actually do the exercise or the movement themselves actively. And so a researcher or a piece of equipment is actually doing the movement for them, say on a treadmill or what have you. And when that's combined with other interventions, time and again, they've seen a massive increase in improvement. And so if you want more detail on another example of that and using training or movement training in neuromodulation and how much better the results are when you combine those two, you can go to our episode with Reggie Egerton, who's just a veteran and legend in the spinal cord injury field, and check out all he has to say about that. I mean, I would say, for anyone who's doing this kind of work, whether it's in the initial discovery phases or maybe after you found some interventions that are promising, please keep in your back pocket that adding some sort of movement training or exercise component to it in general has had additive improvement beyond the intervention alone or exercise alone, and something to certainly consider in your work. So next paper, similar kind of topic. This is from the Animal Models and Experimental Medicine Journal, Talifu et al. Talifu, I always apologize for pronunciation of names in some of these terms. And the title is Effect of In Vivo Reprogramming of Astrocytes Combined with Exercise Training on Neural Repair in Rats with Spinal Cord Injury. Background. The inability of damaged neurons to regenerate and of axons to establish new functional connections leads to permanent functional deficits after spinal cord injury. Although astrocyte reprogramming holds promise for neural repair in various disease models, it is not sufficient on its own to achieve significant functional recovery. Methods. A rat SCI model was established using a spinal cord impactor. 7 days post-surgery, adeno-associated virus were injected to overexpress the transcription factors NeuroD1 and Neurogenin-2, hereafter NGN2, in the spinal cord. The rats were then trained to walk on a weight-supported treadmill for 4 weeks, starting 14 days after modeling. The effects of these interventions on motor and sensory functions, as well as the spinal cord tissue repair, were subsequently evaluated. Results. The combination of NeuroD1 and NGN2 overexpression with weight-supported exercise training significantly improved gait compared to either intervention alone. The group receiving the combined intervention exhibited enhanced sensitivity and sensory assessments. Immunofluorescence analysis revealed increased colocalization of astrocytes and microtubule-associated protein 2 positive neurons in the injury area. These effects were more pronounced than those observed with spinal cord tissue repair alone. Additionally, the combined intervention significantly reduced glial scarring and the size of the injury area. Conclusion. Exercise intervention enhances the pre-programming effects of astrocytes and restores motor function, yielding better results than either intervention alone. All just really, supporting what I just mentioned about keeping in mind adding exercise groups to the work that you're doing in this field. Next, we have from Frontiers in Cell and Developmental Biology via Liu and Tang, Exosomes, New Targets for Understanding Axon Guidance in the Developing Central Nervous System. And this is an abstract of a review paper. I just wanted to include it in case it sparks some interest from anyone who wants to go look at the full-length paper. Abstract. Axon guidance is a key event in neural circuit development that drives the correct targeting of axons to their targets through long distances and unique patterns. Exosomes, extracellular vesicles that are smaller than 100 nanometers, are secreted by most cell types in the brain. Regulation of cell-to-cell communication, neuroregeneration, and synapse formation by exosomes have been extensively studied. However, the interaction between exosomes and axon guidance molecules is poorly understood. This review summarizes the relationship between exosomes and canonical and non-canonical guidance cues and hypothesizes a possible model for exosomes mediating axon guidance between cells. The roles of exosomes in axon outgrowth, regeneration, and neurodevelopmental disorders are also reviewed to discuss exosome guidance interactions as potential clinical therapeutic targets. Very interesting. I want to dive into that one full length myself. Next, we have via Wu et al. In ACS Nano, Metabolic Reprogramming of Neural Stem Cells by Chiral Nanofiber for Spinal Cord Injury. Abstract. Exogenous neural stem cells, hereafter NSCs, have great potential to reconstitute damaged spinal neural circuitry. However, regulating the metabolic reprogramming of NSCs for reliable nerve regeneration has been challenging. This report discusses the biomimetic dextral hydrogel, or DH hereafter, with right-handed nanofibers that specifically reprograms the lipid metabolism of NSCs, promoting their neural differentiation and rapid regeneration of damaged axons. The underlying mechanisms is the intrinsic stereoselectivity between DH and fatty acid binding protein 5, hereafter FAB-P5. I'll use FAB-P5, yep, which facilitates the transportation of fatty acids bound to FAB-P5 into the mitochondria and endoplasmic reticulum, subsequently augmenting fatty acid oxidation levels and enriching sphingosine biosynthesis. In the rat SCI model, DH significantly improved the Basso-Betty-Breshnan locomotor scores over threefold, and the hind limbs compound muscle action potential over fourfold compared with the untreated group, conveying a significant return of functional recovery. This finding of nanoscale chirality-dependent NSC's metabolic reprogramming provides insights into understanding stem cell physiology and presents opportunities for regenerative medicine. So this is really an interesting one. I'd love to hear more about what Wu et al. Are planning to do next with those 3- and 4-fold increases. I guess there's a lot of that in animal models, right? But the question is, how can we be more successful at somehow getting it translated into humans and into the clinic? Okay, our final paper for today from Bioactive Materials via Mu et al. BFGF chitosan brain glue promotes functional recovery after cortical ischemic stroke. Abstract. The mammalian brain has an extremely limited ability to regenerate lost neurons and to recover function following ischemic stroke. A biomaterial strategy of slowly releasing various regeneration-promoting factors to activate endogenous neurogenesis represents a safe and practical neuronal replacement therapy. In this study, basic fibroblast growth factor, or BFGF, chitosan gel, is injected into the stroke cavity. This approach promotes the proliferation of vascular endothelial cells, the formation of functional vascular networks, and the final restoration of cerebral blood flow. Additionally, BFGF chitosan gel activates neural progenitor cells in the subventricular zone, promotes the NPC's migration towards the stroke cavity and differentiation into mature neurons with diverse cell types and layer architecture. These newborn neurons form functional synaptic connections with the host brain and reconstruct nascent neural networks. Furthermore, synaptogenesis is the stroke cavity and nestin lineage cells respectively contribute to the improvement of sensory motor function induced by BFGF chitosan gel after ischemic stroke. Lastly, the gel inhibits microglia activation in the peri-infarct cortex. Our findings indicate that filling the stroke cavity with BFGF chitosan brain glue promotes angiogenesis, endogenous neurogenesis, and synaptogenesis to restore function, offering innovative ideas and methods for the clinical treatment of ischemic stroke. Another very interesting one that I'm excited to hear what comes next for this work. Okay, that wraps up our 5 recently published, this is in January of 2025, papers. This is coming from a selection of papers in PubMed searching for neural regeneration, neuron regeneration, or nerve regeneration. We will do our best to keep these coming so that you guys can stay on top of the newest papers and news and things going on in neuron regeneration. When you're on the go, I know almost everyone in this field is extremely busy, and so I'm hoping that we can, make a mark in helping you to stay on top of this stuff. We also have a fantastic email newsletter that goes out every week. Similarly, we curate 3 to 5 recent pieces of news or papers and put them in your inbox. So again, you're not having to do all that work yourself. We're helping you out with that. So you can go to neuronregeneration.com, scroll down to the bottom of the homepage, or just find in any of the sidebars of most pages, you can join our email news list and have that curated info in your inbox each week. Thank you again for joining me, and I will see you in the next episode.